more osmosis in a cell that has a higher temperature? That's it. Molecules are moving faster. And the faster they move, the more chances to cross the membrane and move from an area of high to low concentration. So this is a perfect illustration of how temperature can influence the rate at which these reactions occur. Nice work, guys. All right. So any other questions about the process of diffusion and osmosis? Does it seem pretty clear? Do you guys feel like you have the terms hypertonic, isotonic, hypotonic in mind? You could predict the direction water moves. You could talk about the difference between animal and plant cells in terms of what happens to them when they're exposed to different concentrations. Yeah?
I just had like a weird kind of thought. I think it was today, or it might have been yesterday. We've been looking at a lot of like diffusion and molecular movement, and we've been seeing it on such a small scale that like thinking about how fast they actually move is actually really fascinating to me, because if someone were to walk in here with, say, like an apple pie, it's like immediately when they open the door, you can smell it. So like seeing it on a scale on a screen is one thing, but then actually thinking about it and how fast like the molecules that give us our experience of aroma like are moving is kind of ridiculous.
Yeah, yeah. And also we are able to detect a really, really small, like we have incredibly sensitive noses, and so we can detect just a few molecules per million for certain chemicals in the environment. So we're really tuned to identify certain things. Like apple pie. Like apple pie, for example. Okay. And I forget, I apologize, but I forget if I showed you this animation or not.
I don't think so.
Okay. I didn't think so, that's why I brought it up, but then as I brought it up, I had this flash in my mind that I'd actually already showed you this. So this is a molecule that is a transmembrane protein, which means what? What's the membrane? Phospholipid bilayer. And we have these proteins that can cross membranes, that can span from outside the cell to inside the cell. And this is a special one called aquaporin. It is a water pore. The name aquaporin tells you a lot about its function. And what it does is it lets water move across the membrane. So water can cross phospholipid bilayers to some degree, but most transit comes through some channels, like this aquaporin here. So if we run the simulation, and I want you to imagine that this is a membrane going this direction, membrane continuing on here. This is outside the cell. This is inside the cell. And what we've done is we've just cut away the membrane so you can watch the molecules of water move, whatever direction they move. And we've painted one yellow so you can track an individual one, because they bounce around crazy, and it's hard to keep track. So I want you to watch the simulation, see if you can see if there's a net movement, and then, assuming this is outside the cell and this is inside the cell, tell me if the solution it's in is hypertonic or isotonic or hypotonic. Got that? All right, let's run it and see. Try it again. What's really cool is these are two amino acid side groups that seem to play a really important sort of, you can think of it as like sentinel role in determining that only water can cross through here. So other molecules, even smaller molecules in water like protons, can't move through here. So take a moment, turn to your neighbor, tell them whether you think this solution is hypotonic, isotonic, or hypertonic.